Chapter 7, Mechanisms of Resistance. The learning objectives for this topic are: list 7 ways by which microorganisms can be resistant to antimicrobial drugs, list 2 ways that genes for drug resistance are spread between bacteria, define cross-resistance, and distinguish it from multiple resistance. Describe 4 ways that development of resistance can be retarded. So the topics for this topic are development of resistance in populations, mechanisms of resistance, multiple resistance and cross-resistance, and retarding resistance. So let's talk about how we get drug-resistant or bacteria. We'll go with bacteria. So in any population before it's ever exposed to the drug, you're going to have mutations. And some of these mutations are going to impart drug resistance to the antibiotic that you're going to expose them to. So in this particular population, almost all of the cells are susceptible or sensitive to the drug, but we've got 2 drug-resistant mutants. So after exposure to the drug, we kill off almost all of the sensitive cells. But we've got these 2 remaining and 1 sensitive cell that happen to be lucky. So this clears the field for more resistant organisms. It's evolution in action. We're eliminating some members of the population, others that have a characteristic that makes them more adaptive to the new environment, spread and become dominant. So this happens all the time. It's a natural process. It's just really annoying when it makes it so that we can't use a drug anymore. So how does resistance develop in these resistant organisms? Well, first let's talk about enzymes that are produced by the microbe. Some organisms produce a beta-lactamase that goes in and clips the lactam ring of penicillin, and this inactivates the penicillin so that it can't inhibit the cross-linking of peptidoglycan. So any organism that produces a beta-lactamase is going to be resistant to the beta-lactam class of antimicrobials, and you have to go with something else. Another thing that the microbe can do is target the drug. What it does is it changes attachment sites for the drugs. So you can change the ability of the drug to attach to the enzyme that the antimicrobial is attacking. So if you change the enzyme, then the drug can't work as effectively on it. So you can slow and prevent entry. The biggest way to do this is to change porins. E. coli can produce porins that are different shapes. Most of the time they're going to have, we're going to call them the large size porins. They have a fairly large diameter, internal diameter. Lots of things can come in and out of the cell. Well, under stress from an antimicrobial, it switches the size of porins that it makes and puts in, say, the small size. Less can come in, but it blocks entry of the antimicrobial. The microbe can also alter its metabolic chemistry. It can produce more of the enzyme that's being attacked, so that even though there's competitive inhibition, you have more enzyme, lessens the effect of the competitive inhibition. Or they can abandon the sensitive metabolic step. Say, for example, with the sulfur drugs. It can abandon the step where it changes PABA into dihydrofolic acid. It just imports folic acid, like humans do, and then the sulfur drugs don't work. Also, there are pumps. These are very annoying. They are transmembrane proteins that as soon as an antimicrobial comes in, it pumps it right back out. It's kind of the revolving door of antibiotics. And resistance pumps have a tendency to pump out more than one class of antimicrobials. It's almost like it's the bouncer at the club. Hey, how did you get in here? Out you go. Now this last one, the MFPA protein, is showing up in drug-resistant tuberculosis, and it's effective against preventing the action of fluoroquinolones. Now fluoroquinolones attack DNA gyrases. These are, helicase is an example of this. These are enzymes that bind to double-stranded DNA, unwind them, and break the bonds. Well, if you prevent that from happening, you can't replicate your DNA and the cell can't divide. Well, some resistant forms of TB have started producing this MFPA protein that binds to the DNA gyrase, preventing the fluoroquinolone from binding, but the DNA gyrase with the MFPA protein can still function. This is the first instance that we've seen where a protein is produced to protect the enzyme without the enzyme itself being changed. Let's talk about the difference between multiple resistant organisms and cross-resistant organisms. If an organism becomes resistant to more than one drug, they are multiple resistant, and we tend to call them superbugs. Now, examples of genuses of superbugs are Staphylococcus, methicillin-resistant Staphylococcus aureus being the most famous. Don't leave out the strep, Enterococcus, Pseudomonas. Pseudomonas has a tendency to be resistant just because it's Pseudomonas. Mycobacterium tuberculosis and Plasmodium, which causes malaria. Now, with the bacteria, they can develop multiple resistance by picking up multiple plasmids that have different R genes. So they're getting resistance because of horizontal gene transfer. Or they can pick up a gene for a multi-drug resistant pump. Cross-resistance, on the other hand, is where an organism becomes resistant to one antibiotic and is now resistant to an antibiotic that has a similar shape. The book talks about if an organism becomes resistant to one aminoglycoside drug, such as streptomycin, that may confer resistance to all of the other aminoglycosides. Another example is penicillin. We have developed semi-synthetic penicillins that have side groups that protect the beta-lactam from beta-lactamases. But you can see how you could get cross-resistance within the beta-lactam group. Even though development of resistance is a natural process, that doesn't mean that we have to encourage it or make it go faster. In fact, we do an awful lot of things to retard the development of resistance. So one of the things that we can do is maintain high concentrations of the drug. We're more likely to kill off all of the sensitive organisms. Your immune system goes in, clears out the resistant organisms. Another method is to prescribe cocktails, more than one class of antimicrobial. That way, the organism that we're attacking has to develop mutations in more than one spot, and that's less likely. We tend to use drugs that work well together. We call this synergism. So when we're making our cocktails, we definitely want them to be synergistic instead of antagonistic toward each other or fighting toward each other. So, for example, you would not want to prescribe a cell wall inhibitor with a protein synthesis inhibitor, because you slow down the protein synthesis, you slow down the growth of the organism, and it's not making as much cell wall, and the penicillin is sitting around saying, well, why did you have me here in the first place? So we want to limit that. We also want to limit use of antimicrobials. We don't want to use antibiotics, save for viral infections, because they're not going to be effective. They're going to eliminate sensitive organisms when they aren't causing a problem. So you only want to use antibiotics when they're needed. We're constantly developing new antibiotics, including semi-synthetic second and third generation. I mentioned that with the penicillins, that we're putting side groups on to protect the sensitive part of the antibiotic. And we're developing brand new antibiotics. We're going to have to do that because, as I said, development of resistance is a natural process. Well, that's it for this topic. I'm including this beautiful summary figure that shows all of the different ways that a bacterium can develop resistance. And here are some reminders of what you needed to learn from this topic.